monohybrid cross and dihybrid cross, we have discussed about the factors that are responsible for the expression of characters as described by Mendel, and now described as genes. We have seen that for any given character, there must be two genes. For example, if you take length of the stem in the pea plant, there are two conditions we have seen: a homozygous condition for tallness, a homozygous condition for dwarfness, and a heterozygous condition for tallness. When it is homozygous tall, we represent it as capital T, capital T. When it is homozygous dwarf, small t, small t. And when it is heterozygous tall, capital T, small t. All these indicating that there are two genes responsible for the expression of a given character. Now it is known that these two genes occupy the same position which is called locus. The position of a particular gene on a chromosome is called locus. So they occupy the same position on the given pair of homologous chromosomes. If you recollect what you have studied in cell division last year, chromosomes are always found in pairs, and in every pair there are two identical chromosomes called homologous chromosomes. Why they are called homologous chromosomes? Because they contain the same amount of DNA and the same number of genes. So in a pair of homologous chromosomes, the two genes responsible for a given character occupy the same position, and these two genes are therefore described as alleles. So two forms of the same gene occupying the same locus on a pair of homologous chromosomes. So on two homologous chromosomes, on one if there is capital T, on the other in the same position if there is capital T, then the plant becomes homozygous tall. If on one chromosome capital T is present and in the same position in the other chromosome small t is present, it becomes heterozygous tall. On the other hand, in the same position in both the chromosomes, if this gene small t is present, it becomes homozygous dwarf. So they are called alleles. Now these alleles sometimes the alleles can modify the expression of a gene. One particular allele of a given chromosome can modify the expression of a gene, or another allele in the same chromosome may modify the expression of a gene. So this phenomenon is called gene interaction. Gene interaction. So gene interaction can be defined as a phenomenon where the phenotype of a given gene can be modified by an allele of the same gene or an allele of a different gene. There are two types of gene interactions. One is called intragenic interaction and the other intergenic interaction. In intragenic interaction, intra, within. In intragenic interaction, an allele influences the phenotype the expression of a particular gene. Its own allele is influencing the phenotype of a given gene. Whereas in intergenic interaction, an allele of a different gene in the same chromosome influences the phenotype of a particular gene. Same chromosome may have different alleles, so one allele of a particular gene influencing the expression of another gene. Therefore, it is called intergenic interaction. For intragenic interaction, we have some examples like incomplete dominance. One familiar example is called incomplete dominance. Another familiar example is called codominance. And we also have in this situation a phenomenon called multiple alleles. So incomplete dominance, codominance, and with reference to codominance, multiple alleles, these are examples of intragenic interaction. Whereas We have examples like supplementary genes, complementary genes, and epistasis as examples of intergenic interaction. So let us first go to the details of intragenic interaction.